So I also did my undergraduate degree at U of T. As you guys mentioned, I did do a double major in human biology and psychology, as well as a minor in physiology. So over my undergraduate degree, I had a lot of opportunity to do research in various environments, in different labs and stuff like that. But during my fourth year research project, it was at Sick Kids under Dr. Amy Wong. I had the opportunity to do my summer research project in LMP, lab medicine and pathobiology, which is my current program. And I had a fantastic experience, not only in that lab, but also kind of in the LMP program and this sort of area of research where you're kind of elucidating a lot of disease mechanisms and kind of investigating different pathologies. So that's kind of why I lean towards applying to LMP for my master's. It's such a fantastic program. It's a bit of a smaller program too. Glioblastoma is one of the most deadly forms of adult brain cancer. And despite the kind of current treatment regimens, which include surgical resection, so kind of surgically removing the actual brain tumor itself, as well as chemotherapy and radiation therapy, the median survival time for patients with GBM is still only 15 to 18 months. So very kind of aggressive cancer, very deadly. And these current treatments have remained pretty much unchanged for decades. And a big problem that kind of contributes to a lack of new treatments and a lack of new therapies for GBM is the ability of GBM kind of cells to develop treatment resistance. So that's kind of where my project comes in. A lot of the current treatments that are being researched right now focus on reducing GBM cell proliferation, so kind of stopping the growth of the tumor. But one really big driving factor in the pathology and poor prognosis for GBM is the ability of the tumor cells to actually invade the surrounding healthy brain tissue. And by doing this, they kind of evade the current treatments that are available. So my research looks at how I can kind of find a therapy that targets the invasive biology rather than just the proliferative biology of GBM, and then kind of finding a second treatment kind of that reduces the viability of the tumor cells, so to kind of kill them all together. And by looking at combination therapies rather than just single drug therapies for glioblastoma, they're much more less likely to develop tumor resistance. So in this kind of way, hopefully will be able to find a treatment that provides better patient outcomes. It's a very aggressive brain cancer, so yeah. This is a good one. A lot of people, I feel like, get pretty interested in the models that our lab uses because we have a few pretty interesting ones. I'd say the less interesting of the two would be what we call spheroid invasion assays. So here I take a clump of tumor cells basically and make them into a sphere. That's essentially like a mini tumor kind of. And then this way I can make like hundreds, thousands of these small kind of spheres and then treat them all with different drugs to test if these drugs can inhibit cell invasion, which is one of the main aims of my thesis. The other model that people seem to really like is called our glioma cerebral organoid or glyco model. This is essentially a cerebral organoid, so kind of like a mini brain, I guess, made from human-induced pluripotent stem cells. And then we co-culture those or combine them with tumor cells, so the actual GBM cells. So this kind of creates a little model, sort of a brain that is that has a tumor. And this way, I can kind of treat these little brain co-cultures with the drugs to see if the drugs that I'm looking at actually inhibit the invasion of the tumor cells through the organoid brain tissue. So it's kind of an interesting in vitro model, I guess, of GBM, and that kind of reduces the amount of in vivo models that we would have to use. So yeah, that's kind of what I've been doing so far, the models I've been using, but we're also developing what's called an ex vivo brain slice model of invasion. So we actually take mouse brains and we kind of keep the brain tissue actually alive and co-culture them with the GBM cells and then we can treat it with various drugs to see if it stops the invasion through the actual mouse brain tissue. And then eventually the end stage would be to obviously test whatever combinations I kind of come up with in mouse models as kind of an end stage in vivo regimen. So that's kind of our workflow right now. So this is actually the first project that I've had the chance to work on in cancer therapy and oncology. So it's been fantastic so far. And it's really shifted my perspective of what I thought kind of cancer research and drug development research was prior to actually starting in my master's. Obviously, there's so many steps between, translating research and translating therapies from benched all the way to bedside. But being part of this process firsthand and seeing the nuance that goes into the research, the steps, the kind of quality control, even the commercialization process of therapies is so technical. And it's really interesting to actually be immersed in firsthand. And yeah, it's really reinforced the idea and importance of cancer research to me, especially for not only glioblastoma, but other cancers as well. There's so much that goes into it behind the scenes in between actually developing therapies and to the point where they can be in clinical trials and actually administered to patients. So, yeah, it's been awesome. Completely changed my perspective, but in a good way. It's been absolutely incredible to conduct research at U of T, which is a fantastic institution, and all of its kind of affiliated institutions within the university itself. And it's really shaped how I conduct my research now. And I'm super grateful to have had the opportunity to work under some fantastic PIs in not only wet lab, but also in dry lab projects, which has significantly influenced my current research approach. I feel like it's given me a better understanding of the entire research process from a molecular all the way to a clinical level, as well as the environment that's fostered at all of these institutions. It's really incredible, and it's provided me the opportunity to learn and grow as a researcher and gotten me to the point where I am at now in my career. I think the kind of choice to do a wider range of programs and kind of broader programs, especially in human biology and psychology, they give you so many options to kind of take courses, I feel like, that you are interested in or you think that you might be interested in. So my background in these majors and even my physiology minor, it was awesome in kind of preparing me for the research process and teaching me about the different, levels of disease that you can look on. I took some very nitty-gritty, like molecular, cellular biology courses, but I also had the opportunity in a major in psychology to work in more of like a human level. So kind of seeing the science all the way across these different kind of fields was incredibly interesting. And even kind of moving into the field of social psychology and more kind of sociological perspectives, I can kind of bring that human aspect now into my research that doesn't necessarily directly work with patients, but hopefully eventually gets translated to that. So kind of a multifactorial outlook on all these perspectives that I've been taught through my majors and minor in physiology. It was awesome. Yeah. So I've played hockey and sports. I've been involved in pretty heavily my whole life, and it's been such a big part of my identity. I think by doing team sports, anyone who's played a sport would know that you learn so much by being a part of a team, or even like extracurricular activities that aren't necessarily sports. You learn a lot of translatable skills that a lot of supervisors and jobs that really look on very highly and respect a lot. My experience in varsity was fantastic. It was definitely very busy, like, being on the ice 6, 7 times a week, and balancing that with the full course load and kind of also extracurriculars and jobs on top of that. But it really did give me a purpose, and a lot of motivation. Time management skills are huge. And yeah, it's been I think it was such an awesome experience to be able to play at a varsity level, have that little varsity community within such a big university as well. So I took a lot of stuff from sports and hockey and really translated it into my everyday life. And I still play hockey too, which is just on a smaller level. So, but yeah, it was such an awesome experience. I feel like there's a couple in this one. Research is full of challenges. So I'd say the biggest one so far that I've kind of found is understanding that science isn't necessarily a 100% perfect thing. And you guys would know this as well. Most of my job every day is troubleshooting and problem solving, which is something that I've definitely learned to embrace and enjoy more as I've gone along. But at the beginning of my research, it was definitely a learning curve, kind of figuring out that your experiments aren't going to work 100% of the time. In fact, they never usually do. So, and learning and to enjoy that kind of just comes with experience. Also understanding that negative results are still results. Even if your results are something that you weren't expecting, or in fact, the complete opposite. I get that a lot too. So kind of figuring out why I got the result that I did, or why this kind of experiment went the way that it did, and being able to be flexible and adapt in my mindset. It's a constant challenge, but it's something that I've really learned to embrace. The solving aspect of research is it's one of the fun parts to me, which is good, but at first it's a bit of a learning curve for sure. So yeah, I'd say that's one of the biggest challenges of science in general. For sure. I think we've kind of touched on this a little bit, but doing research in an area that you love, I think is really important because that can be your motivation every day. And even kind of the opposite of this is not being afraid to explore research in areas that are a little bit outside your comfort zone, or maybe something that, doesn't necessarily align directly with your interests, but could give you great experience nonetheless. You might think that you're dead set in only doing research in maybe stem cells or in cognitive neuroscience, but by branching out, you might find something that you didn't think that you were going to be interested in at all, but you end up really loving. And that kind of opens up different avenues for you as you kind of grow as a researcher and as a person. You still also, kind of on the contrary, even if you end up not being interested in what you're doing as much, you're still developing a skill set that will likely benefit you eventually in whatever area of work that you end up in. I would also say that a big piece of advice is don't be afraid to reach out. I know it sounds super generic, but the worst thing that someone can say, I feel like this applies to everything, is no, not necessarily. They're like, no, we don't have space for you right now in the lab, but reach out in the future. I think there's no harm in branching out and reaching out to as many people as possible. Networking is huge, especially in the science industry. So reaching out to professors early to forge connections, I'd say that's one of the biggest things that I found worked for me. Um, yeah. So this is a big question. I feel like I still at this point don't really know what I want to do. And that's something that I've had to come to terms with, and I've completely accepted it. You guys would know too, you get the question all the time of what do you want to do after you graduate? What do you want to do with this? What do you want to do in science? I think that I definitely still want to do a PhD, like any science kid, maybe considering medicine still, but anything in science where I can, it sounds cliche, but contribute to better patient outcomes and just kind of help people on a bigger scale, whether that be, the research that I'm doing right now or research in different areas that can benefit patients at the end of the day, or drive, important findings in a research area that needs to be kind of conducted. That's something that I want to continue to do. So, whether this be as a professor in industry work, continuing in research in a PhD, medical science liaison is a big one. So being able to kind of communicate my findings and research on more of an industry scale, those are all areas that I find super interesting. So yeah, I'm still trying to branch out and explore even in my master's. I thought that would kind of end in my undergrad, but it never does. I feel like you're always kind of learning and growing. So yeah, that's kind of my end goals right now.